Dear learners, welcome to this online portals of learning. Greetings from the Department of Botany, St. Xavier's College, Palayamkottai. I am Dr.L.Joelri, Assistant Professor in the Department of Botany, St. Xavier's College. I am specialized in molecular systematics. Today, I am going to deal with a very interesting topic called DNA barcodes in herbal drug industries. This is one of the current trend topics that will be more interesting for you to listen to understand the depths of DNA barcodes and its applications in authenticating herbal drugs. Nowadays, people start move to Siddha, Ayurveda, and homeopathic medicine because they trust more in homeopathic medicine or Siddha and Ayurveda rather than allopathic medicine. This is because in certain diseases, allopathic medicines fail. However, Siddha medicine and Ayurveda relies on plants as raw drugs. Now, these plants that are used as raw drugs in these herbal drug industries or Siddha and Ayurveda has to be authenticated. So far, there are many other methods that were adopted to authenticate these drugs. Taxonomic descriptions, chemical identifications, biochemical tests were involved in identifying these drugs. However, the precision of these drugs and their identification is very, very important when it comes to human medicine. Here comes the DNA barcoding and its application in herbal drug industries to authenticate these drugs. Let us move into the PPT to understand what is herbal drug industry and how DNA barcodes are involved in authenticating herbal drugs. Harnessing DNA barcoding for herbal drug authentication. The herbal drug industry has experienced a global resurgence, leading to a surge in the demand for medicinal plants and their derivatives. There are a lot of demand for medicinal plants. We don't get certain medicine. For example, varieties. Osum sanctum is a very common species, but there are other species like Osum gratis, teniflora, which are not easily available. And there is a need for a taxonomist to identify these plants. And there is a lot of demand for medicinal plants in the recent times. However, this rise in the popularity has also brought forth significant challenges, particularly in the area of adulteration and mislabeling. People start to use or misuse these drugs when they are not available. They go for other drugs or other plants and mix it along with this. Now, this causes adulteration, sometimes mislabeling also, that is misidentification of plants. In response to this problem, the scientific community has turned to a very powerful tool called DNA barcoding, a transformative tool that holds immense promise for identifying and authenticating genuine herbal ingredients, ensuring the quality and safety of these precious natural resources. Now, how DNA barcoding is a key to herbal authentication. Molecular plant identification is one of the recent trends. It's called as molecular systematics. Trace out the phylogeny of the plants for identification. Very specifically, we go for molecular detection because plants start evolving very fast, and this evolution leads to a lot of variations. They also undergo a lot of mutations, forming new varieties of species. Recent advancement in molecular biology have revolutionized the field of plant identification. Taxonomy. DNA barcoding, a technique that uses specific genetic markers, has emerged as a reliable and precise method for authenticating plant species. Even at the face of morphological similarities or incomplete samples. For example, you have a plant without flower. Using the conventional method, a taxonomist can identify a plant only with a flower using the Benthamother classification system. Flower is a very important part, or the reproductive morphology is a very important part to identify a plant. We cannot easily guess a plant based on the vegetative morphology alone. Here comes application of DNA barcoding, where you can identify a plant at any stage, even at the seed stage, or at the germination stage, or as a big tree. Or if you have a traces of the particular plant, you can isolate the DNA and identify the plant. Nowadays, plant specimens are identified even if they are stored as herbarium or if they are stored in the formalin. They are able to isolate the DNA using specific methods and identify that particular specimen. Here, the DNA barcoding plays an important role in identifying even powdered mixtures. Now, how it is going to address the adulteration issues? Rise in herbal drug consumption has led to a surge in adulteration because people started to move towards Siddha and Ayurvedic medicine. There comes a demand for these medicinal plants. So what the marketing people does is to go for inferior drugs or substitute certain materials or to use a drug that can replace the genuine medicinal plants. DNA barcoding provides a powerful tool to detect that kind of adulterants and ensure the quality and safety of herbal medicines. Now, this will also pose a threat to the Siddha medicine or the Ayurveda medicine, where some people tend to go for some side effects because of this medicine. Though Siddha and Ayurveda medicine are from original plant specimens, there are certain side effects that are being observed in some people because of the misuse or mislabeling. That has to be avoided, and this issue has to be addressed. Then only people will start to believe in the Siddha and Ayurveda medicine, or else there will be a kind of unbelief or reluctance in the usage of these products. Now, these drugs has to be properly identified by a taxonomist, and then only it should be taken for processing. Now, this is the method, but these drugs are misidentified by the local people, and sometimes it may go lethal also. For example, Brahmi is a common example where it is confused between the northeastern states and the southern states of India. Central Asiatica, in Tamil we call it as Ballare. In southern parts we call this as Brahmi, whereas in the northern parts also they try to confuse it with Bakuba monidi, and another plant, and both these plants are called Brahmi as common names. But both these plants belong to different families. The Centella belongs to Apiaceae, and the Bakuba monidi belongs to Plantageneaceae, where the different families and their different phytochemical composition. But these plants are tend to be confused by herbal drug industries, and this will pose a serious threat and will cause different effect in the formulations. Now, this is one type of example. You can see the plant here. Now, this plant, what you see here is the Central Asiatica. I will try to show it in the pointer. So, this plant is Central Asiatica, where it is a Brahmi, and this plant is Bakapa monuri. Both these plants are confused and they are used misused in the formulations, and both are from different families and both have different category approach. Likewise, there are many examples. Now, here I have just listed a few examples or a few known adulterants. Adulteration is done sometimes mistakenly, but it's done purposely by many other people because of the demand of our plants. This is more threatening, where Piper fratsura can be replaced by Piper valici or other species of the Piper. Piper longum, this tripili, can be substituted by Piper mulisu. Piper nigrum, the black pepper, is commonly adulterated by some of the marketing people or some of the people in the market, herbal markets, as like capsicum annuum can be used or carica papaya. Papaya seeds are adulterated in the pepper seeds. Cider hordifolia is replaced by cider acuta. Solanum nigrum by Solanum melanchina. Terminalia arjuna by Terminalia tomentosa. Now this will tend to decrease the effect of the drug also. Because when you go for a different species, the same phytochemical component may be present, but the effect of the phytochemical component may not be as in the original species. Sometimes the chemical composition also will vary. This can cause side effects. That is why people tend to be reluctant to go for Siddha or Ayurveda medicine instead of allopathic medicine. If this problem or if this issue is sorted, certainly people start to use Siddha or Ayurveda medicine and start to believe in the herbal remedies. So Vithania somnifera can be replaced by Mucuna pruriens and Gingiber officinalis by a local species Gingiber mucuna. Now, what are the advances in the herbal drug authentication of DNA barcoding? So, DNA barcoding is a standardized approach in identifying plants using short DNA sequences. This was proposed by Paul Herbert at the University of Cork. Now it has become very prevalent. However, one loci or one gene or a one intergenic region alone is not enough for authenticating a particular plant specimen. At least two locus or two loci barcoding or a multi-loci barcoding is needed for herbal authentication. For example, you can use barcodes like RPCL, ribulose bisphosphate carboxylase gene, ITS, internal transcriptase, or NATK maturus gene. Now, these are regions that can be classified into genic regions or intergenic regions. Intergenic spaces can also be used. Sometimes spaces like TRN, LF intergenic spaces are also used. However, a combination of a genic and an intergenic region will be more good for herbal authentication in DNA barcoding. So we cover multiple genetic regions known as multi-locus barcoding, which provides more accurate and reliable species level identification when it comes to complex herbal samples. A higher grade of method is high throughput sequencing. Now, this sequencing method is NGS or the next generation sequence technology, where it has revolutionized the field of DNA barcoding, enabling simultaneous analysis of numerous genetic markers and the rapid identification of plant species, even in highly complex mixtures. The recent trend of DNA barcoding is the metabarcoding. The metabarcoding revealing the diversity. See, metabarcoding is the integration of DNA barcoding and high throughput sequencing method, and which is a powerful approach to access the herbal formulations, allowing for detection of potential adulterants and the comprehensive authentication of herbal ingredients. Now, here, metabarcoding is like the metabarcoding in microbes, where microbes are identified, uncultured microbes are identified in petroleum contaminated soils and different other environments where the particular microbe is unknown. Similarly, the unknown adulterants or drugs can be identified and drug can be authenticated using the new method called the metabarcoding method. Let us move into this interesting topic some more, where we use different regions. So I can give you some examples like for Astraci, we go for internal transcript spacing. For APAC, we go for ITS 1 or 2 or PSPA, TRNH, which is an intergenic spacer region. For apocylacy, we go for a specific DNA region. Now, these are all barcoding regions that can be targeted for identification of these medicinal conformants. Analysis, ITS2. Other case, we can use RBCL1. RBCL is a common gene that is used for identifying most of the specimens. However, finding out intergene level variations. ATK gene and RBCL gene can also be used. Another advanced method in identifying herbal powder mixtures is the DNA mini barcoding, and very especially Bar HTM, DNA barcoding coupled with high resolution building analysis. What is this technique? See, you don't need to go for sequencing or hybridization procedures. Just the melting curve of PCR amplicons is subject to the length of the DNA sequence and GC content, and the difference in the base complementarity without going for sequencing or hybridization procedures. Bar HTM monitors the melting curve of nucleic acid in real time by adding intercalating dyes, including the SYBR green, green plus, Riva green, and other dyes. What are the future perspectives? DNA barcoding should be encouraged in pilot DNA barcoding based herbal pharmacovigilance. A routine DNA barcoding authentication to raise the quality and authenticity of herbal registry, giving people more confidence using these drugs, so along with the chemical analytic methods that can be combined, and this facilitate the pharmacovigilance monitoring and signal detection. Now we have a promising field for the DNA barcoding to authenticate the herbal registry. You can post your comments and queries in chat. The queries can be answered later. Thank you all for listening to this short video on DNA barcodes in the herbal registry. Hope it was interesting. Meet you in another video.